Hey, my name is Ryan. I'm a student here at Bon Secours Memorial College of Nursing, and today I've put together a educational presentation on gene therapy treatment. So what is gene therapy? Well, I'd like to start off just to make sure that we're all on the same page of the functionality of our genes. So our cells each contain 46 strands of DNA. A single strand of DNA is made up from 4 different nucleotides. A, C, T, and G are just the letters that the scientists have assigned those. Genes are stretches of DNA with specific sequences of those nucleotides, and genes code for what shape and size proteins are supposed to be made from those cells. And the shape and size of a protein, of course, determines its function. So essentially, genes are the blueprints for your body's protein production. So what is gene therapy? Gene therapy is designed to introduce genetic materials into cells to compensate for abnormalities or to give the cell additional blueprints to make a beneficial protein. So they're all there just to affect the sequences of those nucleotides. And they do so through 3 different methods. They either replace a mutated gene with a copy of a healthy one, they inactivate a mutated gene that is functioning improperly, or they introduce a new gene into the body to help fight a disease. So what is the target for gene therapy? Well, gene therapy has the goal of making the body self-proficient rather than the use of external medical implementations such as blood transfusions or prescription medications. Also, we're targeting those diseases that were previously deemed untreatable, being that we're just enhancing this new technology. And we've also had successes involving all of these different types of disorders, including correcting vision in some blind patients as well. So what are vectors? Well, vectors are very important to gene therapy as they're basically how we're going to be adding these therapeutic gene sequences into our cells. They have 2 different types of vectors: in vivo, which is the gene therapy involving the direct introduction of the vector carrying the therapeutic gene into the patient. They either inject it into or near the targeted organ, or they have ex vivo, which is when a patient's cells are taken out of the body and transduced by the vector in a culture or a petri dish to incorporate the therapeutic gene, and then the gene modified cells are then transplanted back into the patient. So for in vivo, we have the adeno-associated virus, AAV. It is the primary virus that we use for the in vivo introduction. It infects both dividing and non-dividing cells, and it also is very beneficial because it has low pathogenicity. So it means that it does not replicate and kill the cell. And also it's able to target a broad range of specific tissue types because viruses, if nothing else, are very specific in nature to what they target. And also for the ex vivo, we have the lentiviral LV, which is beneficial because we can actually pseudotype this with different envelopes from different viruses. So the capsules in which the DNA is stored in, or the nucleic acid that is stored, can be modified so it can actually even further specify which tissues it is targeting. And also the LV is able to carry more transgenic information, where the AAV is able to carry about 250 to 500 nucleotides. The LV is able to carry about 800, so they're just much larger in size. And here's a picture of the in vivo model where you have the encapsulated virus and is being injected directly into the patient towards the targeted organ. Also, you have the ex vivo model where, in this case, bone marrow is being extracted from the patient, put into a petri dish, introduce the virus, and then put back into the bone. And this is also where they're able to introduce stem cells as well. And here's also a table just showing the various different models of the AAV virus that they're able to use for various different organs. So you can be as broad as you want with AAV9 that can target just about anywhere. Whereas if you would rather be more specific and you're wanting to make sure that whatever genes you introduce are much more contained, you can use AAV4, which will only use the CNS and the heart. And here's also a picture just to show how the viruses actually penetrate into the cell and then also inject their nucleic acid into the gene to modify it. So here's the picture to going through the cell membrane and then attaching itself onto a nuclear pore, and then here it's where it detaches and send all the DNA modification that we stored inside the capsule into the cell. So what is the future of gene editing with CRISPR-Cas9 and TALENs? So with vectors, we discussed the ways that we have to introduce therapeutic nucleotides into cell to help fight diseases, but how does a gene therapy combat the defective gene sequences themselves to remove them? Well, with that, we actually turn to CRISPR-Cas9 system and TALENs, as they are protein-driven genetic editing tools that have the advantage of being able to extract the defective gene sequence while also introducing their own, rather than the viral vectors that are only able to introduce. So TALENs is comprised of a non-specific DNA cleaving nucleotide fused to a DNA binding domain that can easily be engineered so that TALENs can target essentially any sequence. So it can remove and also insert its own sequence, or it can be programmed to, in any stretch of sequencing along the genome. So the TALENs method has already been used to design plants, fuels, pets, and all to our surface specifications. And similar to that of the LV, TALENs method is primarily an ex vivo procedure, so they extract the cells from the patient, modify them, and then insert them back into the patient. And then the CRISPR-Cas9 system, which is actually the preferred method, and I'll get into that later, requires a 3-base PAM sequence in the gene. So of the nucleotides, the NGG sequence is preferred for it to actually extract whatever sequence it needs to and insert its own. So it does make it more limited in where it can replace the nucleotide sequences. But similar to that of the AAV, the CRISPR-Cas9 system is primarily an in vivo procedure, so it's just injected into the targeted area. But CRISPR offers a more widespread and efficient form of RNA cleavage, as it's guided by the Cas9 enzyme, whereas TALENs is, like I said, just much more specific in its target. But CRISPR-Cas9 definitely is the front runner for gene therapy, being as it's the easiest to administer, usually just through a simple injection. But why is bacteria, or CRISPR-Cas9 bacteria's best ally, and why is it preferred in live cells like our own? Well, the CRISPR-Cas9 system originates from bacteria's most effective antiviral system. Bacteria surviving viral infections save a part of the virus's DNA into their genetic code, and they archive it, and that's what we call CRISPR, is that archive. And if the same bacteria is ever attacked again, it'll actually duplicate a strand of RNA from the archived DNA copy that they pulled from the virus, and they will feed it into the enzyme Cas9. So once armed, the Cas9 enzyme will now scan all of the bacteria's DNA for copies of that invader's genetic code, and whenever the Cas9 system finds an exact copy of that nucleic sequencing, it will cut out the virus's DNA wherever CRISPR tells it to, like a pair of molecular scissors, thus saving the bacteria. So this system also works the same way inside our live cells. So while we're hunting for defects in our own genetic code, all we need to do is arm the Cas9 system with both the nucleotide sequence that we wish to replace, as well as the one that we wish to replace it with. And then it'll just go through, and every single time it finds that sequence, it will cut it out and replace it all by itself. So what have been the successes and implications of gene therapy for the future? So the reasons for all this rambling is so that we can lead up to the successes, which are essentially just the why we cares. So to start off with CRISPR-Cas9 and the advancements for HIV treatment. So in 2016, a team of scientists led by Kamel Kali engineered rodents to incorporate the HIV genes into nearly every single cell of their body, which ranged up to 98% of the cells in these rodents actually contained the HIV virus. But using the CRISPR-Cas9 system, the scientists were actually successful in removing the HIV virus in more than 50% of the rodent's body cells just through a simple injection into the tail. So we've actually made huge advancements into not only treating but curing HIV with CRISPR-Cas9. Also, with LV advancements regarding sickle cell treatment, which is another huge treatment, especially in third world countries. So patients with sickle cell disease have mutations in the beta globin generate of an abnormal form of hemoglobin. It results in their red blood cells to appear sickle-shaped. They lack flexibility and they stick to the vessel walls. So in addition to pain, blockage of the blood vessels can occur that slow or stop the blood flow to oxygen to nearby tissues. So they've actually used the LV ex vivo method to remove these cells and then to mutate their genes and place them back into the patient of a 13-year-old patient boy. And he's actually been able to not have to undergo any more blood transfusions for 2 years now, and they're actually moving into larger clinical trials involving 17 patients at the moment to try to advance this practice. And also, so they've actually taken both of these, and with NIH and the Gates Foundation, they're actually doing a ambitious project to bring gene-based treatments for HIV and sickle cell diseases into Africa. I believe at the moment it's raised up to $100 million and towards this charity. And they've announced their collaboration for trials to both cure the HIV and sickle cell within this next coming decade all throughout Africa. And also for the hopeful future, CRISPR-Cas9 is also leading the charge in the fight against cancer because we've discovered with CRISPR-Cas9 that not only correcting defects in the cell is within possibility, but also making other cells more efficient. So with CRISPR, we're actually able to edit our immune cells to make them better cancer hunters. So in 2016, human trials were approved by the U.S. National Institute of Health for researchers to remove T cells from 18 patients, which are your immune cells, with varying types of cancers to be modified and then reintroduced back into the targeted cancer areas. And also similarly, China has joined the team, and they've launched their own research into CRISPR cancer treatment for patients with non-small cell lung cancer. And this work is still in the early stages, but we've seemed to be entering definitely a new age of medicine. Well, that is all I got though, but I appreciate it so much, and let me know if there's anything I missed, and hopefully everyone's walked away a little bit more educated on some gene therapy. Thanks so much. Bye.